UP Board of Regents had a vision to develop their true firstborn college that will spearhead national development through agriculture. At the foot of Mount Makiling, Edwin B. Copeland, together with 7 faculty and 12 students, embarked on the journey to create the UP College of Agriculture, or UPCA. Through its 112 years of existence, the college has produced over 1,000 technologies and variety of plants and animals, 9 national scientists, over 30 outstanding young scientists, and more than 18,000 graduates contributing to agricultural development. UPCA, now College of Agriculture and Food Science, or CAFS, has been continuously contributing to nation building through its instruction, research, and extension function. A mere 10 years after establishment, the college was able to start breeding works with the production of sugarcane varieties with high productivity and resistance to Fiji and mosaic disease. In the 1920s, the importance of agriculture was intensified and UPCA was recognized as neighboring countries began sending their students to Los Baños because of the high quality of education. In the 1930s, breeds with superior characteristics such as the Burkhala pig, Los Baños Cantonese chicken, and Philippine cattle were developed. The 1960s was a fruitful decade for research. Makapuno, a coconut cultivar often made into a dessert, was pioneered and perfected. Meanwhile, the Dairy Training and Research Institute was the first in the country to freeze buffalo semen, paving the way for artificial insemination of local carabaos. Along with the establishment of UPLB in the 70s, the college received the prestigious Ramon Magsaysay Award for International Understanding, Asia's counterpart for the Nobel Peace Prize. In the same decade, the C4 plants were developed. The soil test kits were produced in support of the Masagana 99 program, and the mango induction technique was introduced. Continuing the production of quality research outputs in the 1980s, techniques to minimize post-harvest losses, the accelerated fish sauce manufacturing process, and the rapid test kit for the detection of pesticide residue on vegetables were developed. The 1990s saw many new varieties across species or crop types, including Sinta papaya, the first Philippine-bred hybrid, UPLB Gold, a durian variety, and Doña Amelita, a musaenda named after the wife of former President Fidel Ramos. The award-winning National Corn-Based Farmer Scientist RDE Training Program, or FSTP, was also established around this time and still remains as one of the university's primary extension programs. In the field of instruction, the book titled Integrating Sustainable Agriculture in the BSA Curriculum, the UPLB College of Agriculture Experience, was published in 1999. At present, the College of Agriculture and Food Science, or CAFS, continues to be the premier institution of higher learning in agriculture and food science, and remains committed to nation building through its instruction, research, and extension programs. In order to produce the best graduates, all of our courses are outcomes-based education, and continuous improvement of the curriculum is being practiced to meet the challenges of the 21st century. As testament of excellence, CAFS is constantly recognized by different organizations. In research, we continue to implement relevant studies with focus on the 12 priority commodities to address food security. Moreover, technologies are constantly developed to aid in responsible food production. Supplementing these are extension activities to disseminate the knowledge generated to improve food productivity and livelihood of the people. We believe that present technologies will future-proof our food systems. Some of our notable developments are iDetect, Coraginam, Plant Health and Pest Clinic, Pest Management Tools and Techniques, Smarter Pest Identification Technology or SPTech, Snap Hydroponics, Drone Technology, Rapid Immunofilter Assay for Virus Indexing, Nanofertilizers Fertigrow, Corbogrow, Nanobiosensors, Biochar. We at the College of Agriculture and Food Science will continue to promote and nurture a culture with innovative yet conscientious minds and cutting-edge skills to lead and serve the country and the region. We are one college with one goal towards sustainable food systems.
To our participants from Luzon, Visayas, and Mindanao, magandang umaga, naimbag nga bigat, maaya payabak, marhay na aga, maayong buntag, kaninyong tanan. Welcome to the Coffee Webinar Series. This is brought to you by University of the Philippines, Los Baños, College of Agriculture and Food Science. I am Ricky Brian Biagtan from the Institute of Plant Breeding, and I'll be your host for today's much-awaited webinar episode. We are down to our second episode, and today we're going to talk about development of genetic markers for coffee varieties grown in the Philippines. But before we get started, atin munang alamin ang mga house rules so you know how to participate in today's webinar. For our panelists, kindly keep your microphones muted at all times unless called or requested. For our Zoom participants, you may send your questions through our chat box at any moment during the presentation, and we'll try to accommodate them during the Q&A portion. You may also ask questions after the talk by raising your hand and wait to be acknowledged. For our Facebook and YouTube viewers out there, just type your questions in our comment section down below. When submitting text questions, kindly state your name, the institution you belong to, and your question. So currently, we have 79 Zoom participants and 23 Facebook viewers. So good morning, everyone. So to formally start our program, may we call on the Dean of the College of Agriculture and Food Science, Dr. Elpidio M. Abisit Jr. for his opening remarks.
Hi, Ricky. Do you hear me properly?
Yes, sir.
Okay. Of course, to our speaker today, Dr. Enelia P. Cao, to the Coffee Webinar Committee headed by the Associate Dean for Research and Extension, Lilia M. Fernando Corpus, to all our guests and participants, a pleasant morning to all. Let's rise and grind. It's coffee time. On behalf of the College of Agriculture and Food Science, I wish to welcome you all to the Coffee Webinar Series. For 3 years, the college has been offering relevant webinars for everyone, which are available in different platforms. So wherever you are and whenever you are available, you may access these informations. We are glad that until today, you continue to patronize our webinars and learn from the experts. We are also doing our best in tapping different experts, not just from the college, but from other institutions as well, to make sure that the knowledge being imparted are relevant and up to date. This series, we offer the coffee as a commodity of this webinar because we recognize its importance importance in the country, not just economically, but culturally as well. There was a time when the country was one of the leading exporters of coffee, but due to certain diseases and some certain factors, it declined. For sure, many of us here are also consumers. There are some who drink coffee in the morning before our work starts or after meals to keep our minds going for the day. Moreover, have you noticed that we offer our guests first? It's coffee, right? There's always a significant rise in the number of coffee shops here in UPLB. I can assure you that there are more than 30 shops selling different specialty coffees. That is why we offer this webinar to highlight the importance of coffee and how it is selected and produced to reach our tables. Through this, we hope that the deep understanding of coffee will be spread to everyone. Aside from what we see, the physical characteristic, the distinct taste, we hope to impart the science behind coffee. We hope that you learn a lot today and give more importance to its overall value. To end these welcome remarks, I like to borrow a famous line from famous coffee brand, para kinino kayo bumabangon. May all our dreams and aspirations come true for a sustainable and resilient Philippine agriculture. Hiraya Maniwari. Thank you. Ricky?
Thank you very much, sir. Ayan. So tayo dito sa UPLB, bumabangon tayo para sa magsasakang Pilipino at para sa mamayang Pilipino. Ayan. So thank you very much, Dean, for that very warm welcome to our participants. So before we proceed, let's take some time to take some group photos. To our panelists, may I request you to turn your cameras on? All right. On the count of three, say agree. Agree. One more. On the count of three, 1, 2, 3, agree. Okay. Thank you very much. Without further ado, allow me to introduce our speaker. She was recently appointed this year as a professor emeritus of the Institute of Biology, College of Science, University of the Philippines, Diliman, Quezon City. She has served the university for 42 years, rising from the ranks from instructor to full professor. She earned her bachelor's degree in biology, major in genetics from the University of the Philippines, Los Baños, her master's and doctor of philosophy degrees in biology, major in genetics from the University of the Philippines, Diliman, with a sandwich program at the University of North Carolina, Chapel Hill, North Carolina, USA. Her leadership roles within the university include serving as the former director of the Institute of Biology, as well as the Natural Sciences Research Institute at UP Diliman, and as the deputy executive director of the Philippine Genome Center, UP System. Beyond the university walls, she actively contributes to the field as the vice chair of the Department of Agriculture Biosafety Committee, a member of the Department of Agriculture Technical Advisory Group on Modern Biotechnology and Other Innovations for Agriculture and Fisheries, and also a member of the Technical Panel for Biotechnology, Genomics, and Nanotechnology of the Philippine Council for Industry, Energy, and Emerging Research and Development, or DUST PSERD. Her research focus, particularly in coffee genomics and cyanobacterial biotechnology, is well reflected in numerous articles published in Scopus Index journals. She has held prestigious positions as the former president of the Philippine Society for Microbiology Incorporated and the Philippine Society for Cell Biology Incorporated. Currently, she serves as the Senior Ambassador of the Philippines to the International Society of Microbial Ecology. Noteworthy recognitions in recent years include the William L. Fernandez Excellence in Teaching Microbiology Award and the UP Scientist, both bestowed upon her in 2021, along with the Filipino Faces of Biotechnology recognition in 2019. To enlighten us on the development of genetic markers for coffee varieties grown in the Philippines, let us all welcome Dr. Ernalea P. Cao.
Good morning to everyone. First, I'd like to thank the College of Agriculture and Food Science through Ma'am Leah Fernando for inviting me to present this talk today. Let me share my screen first so I can also show you my slides. Let me put these in slide mode. All right. So the talk that I will be presenting today is actually a compilation or summary of the different researches or work on coffee that I have done through the years. I have included in this talk the many colleagues, my former thesis advisees, research personnel, and collaborators from the different universities. So let me also give to you the outline of my presentation. So this will be the components of what I'll be discussing with you today. And to start, coffee is said to be the world's second most traded commodity, the first being crude oil. And the Coffea species belong to the family Rubiaceae and accounts for 124 species. Of these number of species, 3 are used as beverage, and these are Coffea arabica, Coffea canephora, and Coffea liberica. This slide shows us the global coffee production, whereby we can see that Coffea arabica, known as Arabica coffee, is said to produce the highest amount because it is very popular because of its good taste and aroma, followed by Coffea canephora, also known as Robusta. In the Philippines, 2 additional varieties of Coffea liberica are cultivated, and these are varieties liberica or Baraco and De Webre or Excelsa. Now, the concept of genetic markers can be traced back to Gregor Mendel when he examined differences in parental and offspring phenotypes and eventually genotypes in garden peas. So this gave rise to the origin of what were then known as variants. And in the case of Mendel, he used characters for genes which served as what we now call as genetic markers. The variant allele that is used to label or trace a biological structure or process is then referred to as a genetic marker. Through the years, the use of the concept or the word genetic marker has changed or has expanded. In the case of coffee, the identification and characterization of genetic markers in coffee will serve as tools for preserving the genetic identity of the local varieties, as well as to determine which to plant or grow. At the same time, assurance of genetic consistency is also important in any product to ensure that the same material is used, particularly in the case of coffee blends, where mixtures are utilized in order to combine the best characteristics of different varieties. So in this case, we can trace from what variety or varieties the blend would be made up of. And selecting diverse genotypes will also be useful for breeding programs for the genetic improvement of coffee. Now, these were the objectives of the studies that I had conducted through the years. At the start, we wanted to determine the identification of reference and unknown varieties because there had been some mislabeling also or misidentification. Second, we wanted to examine the genetic relationships among the coffee species and varieties. And then thirdly, we wanted to identify markers for traits that are needed by the industry. And subsequently, as an application, we would like to develop a detection system for these important traits. Now, the first marker that we tried to study would be morphological in nature, but this time we used a particular type of morphological examination, which is referred to as morphometrics. The word comes from the Greek morph, which means shape, and metron, which means measurement. And there are 2 types of morphometrics, the traditional and what is known as the modern geometric morphometrics. When we speak of traditional morphometrics, this utilizes linear and angular measurements as well as meristic counts and uses multivariate statistical methods to arbitrary collections of size or shape variables such as distances and angles. In the case of modern geometric morphometrics, then this utilizes certain what we call as landmarks, which are specific biological points with some developmental, functional, structural, or evolutionary significance. We examined leaves and used traditional and modern geometric morphometrics to study variations in the leaf characters. For meristics or traditional morphometrics, we, let's say, measure the maximum length of the leaf, the petiole length, blade length, maximum width, the length above the maximum width, and the length below the maximum width, and used ANOVA as a statistical tool to examine the variations, also using a particular software, which is the SPSS. For modern geometric morphometrics, which again is landmark-based, so an example would be, let's say, this leaf from Coffea arabica. So we took pictures of different leaves. We took the mature leaves, which would be the third leaf from the base of each plant. We collected 95 leaves from each of 95 varieties and placed landmarks such as this. And so these were placed along the margins, and then the pictures were stored in a computer, and then we used a software whereby the digital image was stored, and then we used GPA to superimpose the landmark coordinates as shape variables. And then we used a program called the Integrated Morphometric Package in order to analyze the shapes of the different leaf samples. This is the table that shows us the different morphometric characters as measured in the four different varieties. So this would be Coffea arabica, liberica, robusta, and Canephora, and Dewevre or excelsa. And you can note that the morphometric analysis of the leaf measurements indicated significant differences in length and width among the four varieties, as can be shown by these representative examples, whereby Arabica had the smallest leaf length, while Canephora had the largest leaf length. And then you can see differences also in the width of the leaves of each variety. So whereby, say, for example, excelsa show the widest width, no, or the highest width, and Canephora showed the longest leaf length. Now, we also used pollen grains to study morphometrics, again, using the representative varieties Arabica, Canephora, Liberica, and excelsa. And under the microscope, we examined the view of the pollen from the polar end, polar view, and at the equatorial end. And we measured the following characters: the colpus with, which would be this groove here, no? So this is, let's say, colpus 1 with, colpus 2, colpus 3, and then we also got the average colpus width with the per pollen and the pore diameter, which is seen here. And then at the equatorial view, we also measured the polar diameter, the equatorial diameter, the endocolpus width, the endocolpus height, and the pore diameter. Again, using the same statistical tools as we have used in the leaves. And then we also placed landmarks along the polar and equatorial view, 12 landmarks in the case of the polar view and 14 landmarks in the case of the equatorial view. Again, took pictures, digitized them, and then used the TPS Dig2 software in order to store the images along these landmarks. And these were the descriptions of the landmarks for each sample. That was the polar, and this was the equatorial view. And then this table shows us the characters again that were measured in the 4 varieties that were examined. And to summarize, we found that, as we have seen earlier, the leaf length of Arabica was significantly smaller than the other varieties. Canephora had a more similar shape to Arabica, well, because Canephora is one of the parents of the tetraploid Arabica. We found also significant differences between the 2 varieties of Liberica, which supports the earlier study of Kumar et al. In 2008, which placed Liberica as a distinct variety. Excuse me. The pollen morphology variations also support and are consistent with the findings on the leaf morphology and the DNA sequence results, which we also conducted and which I will be discussing in a while. From these studies, we were able to publish our results for the molecular and morphological differentiation of the Coffea varieties and the pollen morphometrics study. Now, another character or 2 characters that we studied for morphometrics were the flower and fruit morphometrics. So again, we measured, let's say, the length of the pistil, the length of the corolla. We counted the number of petals and so on. And then for the fruit or the berry, we measured, for instance, the maximum longitudinal length, and then we also viewed again the polar view, the major radial axis and the minor radial axis. And then we also conducted geometric morphometrics, placing landmarks, let's say in this case, the corolla tube, and also again landmarks on the longitudinal and radial portions of the berry. These are again the measurements that we got for the flowers and then the fruits. And these were the findings that we got. Arabica can be easily distinguished from the other varieties on the basis of the corolla length, petal shape, berry shape, and length of its radial minor axis. Canephora, on the other hand, by its higher number of petals and stamens, corolla length, longitudinal berry shape, petal shape, and length of its radial major axis. Liberica, on the other hand, is recognizable by its corolla length, longitudinal berry shape, and length of its radial major axis. And the Wevre can be identified solely by its metric characters such as corolla length and radial major axis. Again, we were able to publish our results from this study. Now, another set of markers that can be used to differentiate coffee, particularly in this case, the 2 varieties, would be in the case of biochemical markers. Now, coffee has been attributed with certain bioactivities of the seed extracts. And what we did this time would be to use the lesser studied parts, which would be the leaves, anyway, are just shed off and discarded, and tried to collect Liberica and Excelsa samples from two places in Cavite and Benguet, and then also segregated the immature leaves, the ones that are found on the tips, and the mature leaves, the ones that are found at the base of the plant. And using two extraction procedures, methanol and simply water, we tried to conduct antioxidant testing, antimicrobial testing, and cytotoxicity testing. The antioxidant activity was measured using the DPPH assay, while the antimicrobial activity was determined using the Kirby-Bauer method. These were the results that we got for the antioxidant activities of the different leaf extracts. We used ascorbic acid as our control, positive control, and we found that in this case, the Excelsa leaf extracts have the highest antioxidant activity compared with the Liberica leaf extracts. Methanol was the more efficient extraction solvent compared with water. And while the correlation of the age of the leaves is not very clear, results showed that the mature extracts are generally more potent than the immature extracts, perhaps because the mature leaves have already accumulated enough antioxidant compounds compared with that of the immature leaves. Again, we were able to publish our results from this work. So the idea here is that aside from having coffee from the beans in the berries, we could also probably then have tea from the leaves of these coffee plants. Now, this table shows us the average measurements that we got for the zone of inhibition of the different extracts, the control that we use. So this diffusion results for the Liberica extracts and the diffusion results from the Excelsa extracts. The antimicrobial activity of the Liberica extracts and the antimicrobial activity of the Excelsa extracts. To summarize, again, the Excelsa variety showed a greater antimicrobial activity than the Liberica extract, the Liberica extract or variety. The methanolic extracts, again, were found to have a wider range of antimicrobial activity than the aqueous extracts. But this time, we found that the immature Benguet extracts, even of both the Excelsa and Liberica, were found to have significant activities as compared with the other leaves, the mature leaves. And this time, it is possible that the immature leaves would have developed a more effective way of resistance against microorganisms. Now, let's have a coffee break first because I will be discussing other studies that we have conducted on coffee. Maybe for this just particular trivia, as we can call it, you can recognize and probably name or identify the celebrity holding coffee cups. So on our left, you probably can identify her. She's Angelina Jolie, and on the right, she is Sandra Bullock. Okay, so let me continue. So this time I'll be dealing with molecular characterization. And why is there a need for molecular characterization? So we were actually doing then morphological, biochemical, and molecular characterization at the same time. And why is this so? Because we note that molecular characterization will complement the morphological and biochemical characterization of the different varieties. Molecular characters or markers are more stable than morphological markers, and these characterization again will serve as tools for preserving the genetic identity of our local varieties. So we use different molecular markers. The first one were universal DNA barcodes for plants. These were the MATK or maturase and the RBCL or the Rubisco genes. They are found in the chloroplast and are said to be stable and highly conserved in plants. That's why these are the universal barcodes that are utilized for plant molecular taxonomy and systematics. And so what we did was to design primers for these genes. These were the sequences. And then did PCR. These were the running conditions. And then excised the bands that we produced for each gene marker and subjected them to sequencing. Afterwards, the sequences were subjected to a software whereby we were able to come up with phylogenetic trees or using a neighbor joining tree. This is the phylogenetic tree that we got for the MATK gene sequences. So we also analyzed, aside from the samples that we got, specimens or Coffea sequences that were available in the GenBank database. So as you can see, the Arabica formed a distinct clade. This was actually true also for the RBCL genes. And then this time, the Liberica and the Excelsa varieties grouped together. And then, of course, the other species that were found in the database then grouped separately. In the case of the RBCL gene, we just wanted to focus particularly on the Liberica and the Excelsa varieties. As you can see, again, they formed one clade. And so we found that using these DNA barcodes, we could not distinguish between the two varieties. And well, maybe this would also be consistent and will support the work of Davis et al. In 2006, whereby, yes, Excelsa or de Vevre is a variant of Coffea liberica, but they still belong to the same species. That's why probably then they are very closely related. Now, we then collected for more samples, not only the samples that we got from Cavite, we also collected NSIC or National Seed Industry Council registered coffee varieties from the Bureau of Plant Industry in Benguet. These varieties we now tried to compare also and characterize. So these were the samples that we collected from Cavite State University in Indang, Cavite, and from the Bureau of Plant Industry in Baguio City, Benguet. And we also examined the RBCL and MATK genes for this. And we found, indeed, identities to confirm that these were Canephora species, this Robusta varieties, this is Liberica, and these are Arabica. They belong to Arabic varieties, or species rather. Just to show you the neighbor joining trees again for the MATK and RBCL trees. Again, the Arabica samples clustering together as contrasted to the Robusta and the Liberica samples. So we also used random amplified polymorphic DNAs as another molecular marker. So we used 4 random decamers, designed these primers, and used them again for PCR. These were the running conditions, the components. And then we scored the RAPD patterns using the NACE genetic distance, using the software PubGene. These were the banding patterns that we observed. We had 5 replicate plants for each variety and each primer that was used, and the genetic distances for each one. As you can see, then this would be the scores would represent the genetic relationships that are more similar here. These would be, of course, more similar, and then more distantly related in the case of these other combinations. The dendrogram that was produced, again, showed distinct differences in the Arabica, Robusta, Excelsa, and Liberica varieties. Now, so we wanted to focus on species and varietal differentiation that would be associated with certain characteristics, such as resistance to pests and diseases, and then later on, also those that would have something to do with taste and aroma, and also including yield. But I shall be discussing pest and disease resistance for this particular talk. So if we compare, Arabica is said to be more susceptible to pests and diseases. Canephora is more resistant compared to Arabica, same as Liberica. But we want Arabica because it is the species that contains the varieties that has good taste and aroma. So we compared, again, not only the NSIC registered varieties, but also those other Arabica varieties that were available at the Benguet State University in Benguet, and then also compared the Robusta samples from Benguet, also with those that are found in the Cavite State University, those that are found in Calamansig in Sultan Kudarat, and Malaybalay, Bukidnon. And then, of course, we had the Liberica samples from Cavite State University. These were identified in the specific places or universities that or from where we have collected the samples. So what we did at first is to use published markers in the form of SSRs, or short, simple sequence repeats, or microsatellites, which would, as the word connotes, short repeats that are distributed throughout the genome of plants. And so the number of repeats that are present can also differentiate then each species. And so, as mentioned, we used 20 published SSR markers from the work of Teresa and co-workers, and tested these markers. Now we synthesized the primers for each one, did again PCR, and tested if these primers can differentiate the varieties that we were studying, the local varieties planted. And this represents the neighbor joining tree for the different samples that we collected. And as you will note, the Arabica, again, samples grouped together, but there were different groupings, let's say, from those that could be found in the SU compared with those that are found at the Bureau of Plant Industry, as you can see also here. And then also, however, those that can be found in the other places. This focuses on the Robusta samples that we collected. And as you can see, it seems that those that are found in the same place would group together the BPI Benguet samples as compared to the Sultan Kudarat samples, the Bukidnon samples, and the Cavite samples. We also found certain SSR primers that can distinguish the varieties. Like, say, for example, this SSR primer 124577 showed distinct banding patterns for the Arabica varieties. However, you note that Red Bourbon showed a different banding pattern compared with Yellow Bourbon and Catura. According to our collaborator in BSU, this may be just the same variety. The Robusta varieties also showed distinct patterns. They had the same patterns here because these are clones planted at Cavite State University. This is another Robusta variety that also had the same pattern, and Liberica also having a different pattern. Another interesting marker that we found was SAT235, which we noted would be having this band that would be present in the Robusta varieties, but would be absent in the Arabica varieties, as well as in the Liberica variety. And this seems to be consistent with the report that this marker is associated with disease resistance against coffee berry disease, and whereby it would be the Robusta varieties which are resistant to such disease. And we use also other primers to further differentiate the varieties, and came up with papers also reporting our results for specific aspects of our studies. Then we went on to try to develop a detection system for two interesting pests, the coffee white stem borer and the coffee leaf rust, for identifying which would be the resistant and which would be consequently the susceptible varieties. Well, because we know that our local coffee production is still hampered by pest and disease infestation. And these are the many pests and diseases that plague our coffee. And however, as I've mentioned, we are focusing on two, one pest and one disease. Now, why did we want a detection system? Well, so that we can determine which are genotypically resistant and or susceptible, which will aid coffee growers or farmers and even researchers in planting only those that can withstand infections. And at the same time, maybe later on, these markers can be used in marker-assisted breeding programs for the genetic improvement of coffee, whereby, let's say, the resistance genes can be transgressed to certain varieties of interest. So this is the coffee white stem borer. The pest is due to Xylotrichus quadripes, and it had been found from previous studies that there is a gene that codes for an enzyme, the pectate lyase gene, that will be responsible for breaking down the borer or the insect. So this has been studied, mapped already, so we could identify that gene. So in this case, the marker was actually a gene. And for disease resistance, as you said, we wanted something that could identify a resistant and or susceptible plants to coffee leaf rust, because this is a major cause of infestation diseases in coffee. There had been studies also on the genes that are said to control resistance. Of course, there are also virulence genes on the other side of the coin, which would be the pathogen. As we know, there is that host pathogen interaction that is also taking place. For this detection system, we used what is known as loop-mediated isothermal amplification, or LAMP, which utilizes at least four primers to recognize 6 distinct regions on the target sequence. So either the resistance sequence or the susceptible sequence. Now, we wanted to use a non-PCR based detection method so that this can be done even in the field. As we know, PCR based methods will need a thermocycler that will be present or used in the laboratory. So what we did was first to search for potential LAMP targets. So we did an extensive bioinformatics analysis to search for sequences that can be used for LAMP, which had been established and validated. So we also had publications on the bioinformatics survey of this candidate resistance genes, mapping of these genes, and so on and so forth. And so we collected again leaf samples of Arabica, Canephora, and Liberica coming from different places. We would like to emphasize also on Liberica because this is what we would like to develop also in the Philippines as specialty coffee. Of course, after collection, we stored the samples and then eventually did DNA extraction, designed and synthesized primers. So these were the primers that we initially tested for CLR. These are supposed to be scar markers this time, sequence characterized amplified regions. And the primers this time for the coffee white stem borer. So we also had the 3 primers designed. And then we optimized first PCR conditions. So we tested first for the presence of the markers in the laboratory. So these were the PCR conditions for CLR, the PCR conditions that were optimized for CWSB, just to show you how the experiments were conducted in the laboratory. And then after sequencing, we again, PCR we excised the bands and sent them for sequencing. And then we got trace files, examined the trace files to see if there were differences in the sequences in the different primers that we tested. And then from there, we converted now and designed primers, converted from the PCR-based primers to the LAMP-based primers using certain softwares. So these are the results that we got for this particular marker, which we eventually chose, the BA12412K F marker. As you can see, there is a lower band that is present in all the samples or the varieties that we got, and which is said to be from literature reported to be resistant. So we were surprised that, well, even the supposed to be non-resistant or susceptible varieties contain this. But what was different is that we got also a higher band, about 400 BP, which was only present in the susceptible Robusta, Arabica varieties. And so what we did was to excise both the supposedly resistance bands and the susceptible bands, had them sequenced, and then designed primers, particularly for supposedly this resistance band, and then also primers for the susceptible band, for the one in the upper region. So eventually these were the LAMP markers that we synthesized. These ones shaded in yellow were for the resistance marker, and the ones shaded in blue-green were for the susceptibility marker. And so upon testing from the PCR results to the LAMP, which is a colorimetric method, the substance in the mixture is originally pink in the absence of any, in this case, because this would be for the resistance marker, there's no template. This is just all the mixtures except the coffee DNA. So it did not change color, and then the rest would change color. So we had to also optimize the conditions for incubating, no? And these were the these are actually replicates, no? For the susceptibility marker, as you will note, so there are no susceptible bands in these samples, and it turned out that the susceptibility band was detected in a yellow bourbon and Caturra. It seems from reports that red bourbon somehow develops probably resistance to coffee leaf rust, and may be interesting in our studies also to recommend this kind of variety compared with the other Arabica varieties. For coffee white stem borer, just to show you rather, again, consistent, different banding patterns in PCR for yellow bourbon and Caturra compared with the rest. This is for the coffee white stem borer. So the resistance gene seemed to be present in all, but we sequenced each band anyway to check if there could be differences in the sequences. However, the band size may be the same, but there may be differences in the sequences. And so these were the LAMP primers that we designed eventually for the pectate lyase gene. The results, again, as we can see, this time the Robusta, rather the Arabica varieties were susceptible, and the rest are found to be resistant to coffee white stem borer. The PCR results replicates done by 2 people just to see if the results were consistent. And then we also did RNA expression analysis just to check. So let's say for coffee leaf rust, so we found also the susceptible band being expressed in these varieties, yellow bourbon and Caturra, during infection only. And then the resistance bands being present also in all. And then for coffee white stem borer, we saw also the presence of the expression of the RNAs here, except for typica, which is found to be showing rather the expression also, and no expression in the uninfected plant. So at this point, we're still conducting field testing to check other varieties, unknown, let's say, varieties. We are testing this together with our collaborators at the Cavite State University. And eventually would like to come up with a kit that could be used by farmers or plant growers. I'd like to acknowledge the funding support of the Department of Agriculture Bureau of Agricultural Research, which did funding for the morphometrics part, the original, the first rather researches that we did on coffee, as well as the biochemical characterization. And then later on, the Department of Science and Technology, Philippine Council for Agriculture and Aquatic Resources Development for the funding of the molecular markers aspects of the projects. The Philippine Genome Center and the Institute of Biology for the use of the facilities and support also. Thank you very much for your kind attention.
Thank you very much, Dr. Cao, for that very informative talk. Allow me to acknowledge some of our participants. We have participants coming from the Apayao State College. We also have from National Teachers College, Isabela, Ifugao State University, Hapao Campus. We also have participants from the Eastern Samar State University, University of Science and Technology of Southern Philippines, Claveria, Mindanao. We also have from Occidental Mindoro State College. Also from Bontoc, Southern Leyte. We also have participants from the Batangas Coffee Farmer Federation. From Davao City, University of Antique Hamtic Campus. We also have participants from the Palawan State University, Iloilo, Osamis City, Misamis Occidental, from the Regional Office of Department of Agriculture in Region 10, Field Operation Division. We also have from Tacurong City, Sultan Kudarat. We also have from Norala, South Cotabato, Davao del Sur State College and Mindanao State University Main Campus. We also have from the Arellano University, Mandaluyong City, Cotabato Foundation College of Science and Technology. Yan. Good morning po sa inyong lahat. We also have from the De La Salle Araneta University. Good morning po. From the Pago Oriental Mindoro Entomology Laboratory of the Institute of Plant Breeding. Good morning to all of you. We will now proceed to take some time for your questions, for the questions coming from the participants. As a reminder, you can still submit your questions via chat box and comment section or just raise your hand and wait to be acknowledged. I will now yield the virtual floor to our moderator, Ms. Mads Purificacion, a university researcher from the Institute of Plant Breeding.
Okay.
Ms.
Thank you.
Mads, take it away.
Entitled Development of Genetic Markers for Coffee Varieties Grown in the Philippines. So this presentation is a culmination of all the studies done by Dr. Cao's team. So she defined what genetic markers are and how it can be used as tools for identification and characterization of coffee. Okay, so the floor is now open for questions. And again, may I encourage everyone to ask questions, and you can raise your hand or type your questions in the Q&A button. Kindly state your name and affiliation when called. For YouTube and Zoom participants, you may also type your questions in the comment section and we will read it later. Okay? So, ako po muna, no, habang wala pa pong nagtatanong from our participants. So, Dr. Cao, hello po. So, very interested po ako dun sa detection system nyo. Since here in UPLB, we're also developing a kit for disease detection naman po, no? So, ask ko lang, ma'am, ano po kaya yung range ng price nung kit po, since if we were gonna use it for marker-assisted selection po, or marker-assisted breeding, medyo madami po yung samples. Lalo na po kung, yun po, kung gagamitin po namin siya. How much it cost po, and sa isang kit, ilang samples po kaya yung kaya nang i-detect for resistance screening? Yun po.
Thank you very much po for the question. Actually, we have not yet done an extensive parang economic kung ano ba analysis of how much, no, if ever the kit will cost, kasi yun nga, we're still doing validation. Pero during the proposal stage, we were asked also to estimate based on the price of the kit and then how much we can parang use for each sample. Yung na-estimate namin, about less than 200 pesos ang magagamit, parang yung cost for testing, let's
Per
say.
sample.
Per sample, yes, yes. Oo, kasi parang that is why Mona, who is also here with us, yung aming DOST research fellow, tried to optimize the conditions for the lamp assay na ma-reduce yung parang kung ano yung pinaka-smallest amount of the lamp mixture that we can use para magpakita pa rin ng positive results or negative results. So because we want to keep the price low. So yun nga, less than 200 yung na-estimate namin sa proposal. But we'll see as we field test nga or validate kung kaya pa rin. Kasi what we did was to first use nga yung mga NSIC varieties or from our collaborators, kung hindi man sila NSIC registered, identified nila na resistant or susceptible para matest namin yung kit kung magpapakita nga ng positive or negative reaction. And then what we're doing now is to field test nga yung mga coffee samples from farms, supposedly na observation lang ng farmers or ng growers sa CAFS na ito nagkakaroon ng leaf rust din or ito hindi nagkakaroon. So we'll see. Sana we will be able to come up with cheaper pa nga na price when we develop the kasi parang ang idea namin in the last slide, parang kit siya talaga na we will give tubes and then on the field. Ang isa lang na kailangan na ma-optimize yung, kasi we still have to do DNA extraction in the laboratory. So, yung sana, yung idea ko, yung if you put the leaf in the mixture, parang ma-extract na yung DNA and then nandoon na yung reaction so that everything is really field-based. Yun yung pinaka-goal sana nung the kit development is another kung ano ba phase of research na hopefully matutuloy ng mga gumagawa ng coffee researches pa rin.
Okay po. So thank you Dr. Kao. No, actually sa ano din po no, medyo malaking gap po talaga yung DNA extraction from the field. So yun din po yung challenge po sa amin naman po for plant pathogen detection po.
Oo. At saka yun nga pala, if I can just add. So ang usual na mga kits nga detects the pathogen. So let's say in coffee leaf rust, the presence of the fungus, let's say Himalaya, Bastatrix. Yung sa amin, yung plant sana yung dinidetect, yung genetic kung ano ba identity ng halaman. Oo. So medyo challenging then because we know disease resistance is actually a complicated, a complex trait. Madali nga sana yung pathogen kasi present or absence. Kaya lang, why are we doing the detection in the plant? Kasi parang what we wanted sana is to determine resistance even before the infecting agent comes in. Parang yun yung concept ko na bago magkaroon ng fungus or fungal infection or berry borer infection or yun nga stem borer infection, alam natin kung magre-resist siya o hindi. Yun lang. Thank you.
Thank you so much po Dr. Kao. So we have a question po here in Zoom. So from Matthew Sino, good morning po, a home brewer from Northern Coffee Community po. An icebreaker question po at the end if possible. Ano pong variety ang paborito nyo po na i-brew or inumin, Dr. Kao?
Actually, well, parang ironic nga kasi I'm not really a coffee drinker in the sense na meron akong mga thesis advices noon na talagang sila kung ano ba yung coffee connoisseur, ika nga, no? Yung
mahilig talaga, and they know the taste, no? Pagka pumunta kami sa coffee shop, yan, alam nila, ganan yung kung ano yung ito, sabi niya, ito may barako, especially yung blends kasi nagtatry kami iba-ibang blends. But personally, actually Arabica is really good, no? But I also like yung barako coffee. Yung kick that it gives, gusto ko. And I think yung maybe that is what we can do also in the future, yung blends, o kaya introgressing or transgressing, let's say, the resistance genes to other coffee varieties. Kasi ang problema lang ng Arabica, medyo susceptible siya. But if I were to choose a variety, I like red bourbon.
Okay, so red bourbon daw po, no? Okay, ma'am, we have another question po. So from Marlo Salinas, good morning po. Just a point of clarification po, did you establish the identity of coffee varieties as to Robusta, Arabica, and Liberica based on SSR markers only? Thank you.
Thank you po for the question. So actually, ano po, we have a lot of work on SSRs, but as I have shown, gumamit din kami ng ibang mga genetic markers like yun ng MATK, RBCL, even yung RAPDs, and even the morphological markers, especially leaf morphometrics, na identify namin na iba yung Arabica talaga. May distinct siya na grouping kung ano ba dun sa phylogenetic trees. And then yun nga, we wanted to differentiate yung mga varieties kasi ang dami-dami palang varieties ng Arabica. Kaya lang yung mga sikat kung ano ba in quotation marks, yung mga NSIC registered, yung ng Yellow Bourbon. Yung Katura at saka Yellow Bourbon, sabi nga nung collaborator namin from BSU, they think it's just the same variety. But Bureau of Plant Industry parang classified them separately. So yun. And then yung Robusta, actually, parang isa yung klase lang nung Robusta na nakuha namin na yun nga, yung NSIC registered from CAFS, pero meron din daw yung planted ng Nestle in Mindanao. At the same time, yun, yung Liberica na di-differentiate namin yung Barako at saka Excelsa. But in some markers, as we have shown, hindi sila ma-separate. Maybe because they really belong to one species. So, ganun po yung pagka-characterize namin or identify. I hope I answered the question clearly. Okay po ba?
Mukhang okay na.
Ma'am.
Kay Ma'am Marlo po.
Oo.
Okay. Okay, wala pa po siyang follow-up question po ulit.
Sige po.
Okay daw po. Salamat daw po, ma'am. Okay po. So, next question po. So, from Bernard Panabang. I am interested in host plant resistance breeding against insects of coffee, but we do not have coffee projects in my institution nor germplasm collection. Can you point me out which institution is ideal for pursuing a career in HPR for coffee, or are there possible collaborations that can be done?
Based on our experience, so Cavite State University nga in Indang Cavite maintains a germplasm collection of coffee, especially because it houses yung National Coffee Research Development and Extension Center, yung NCRDEC. Aside from Cavite State University, yung Benguet State University in Benguet, no, also has an extensive collection of other, aside from the NSIC registered, yung ginamit namin from our collaborator kay Professor Macanes, na iba-iba ring varieties of coffee. In Mindanao, may work din on coffee yung Sultan Kudarat State University through ang collaborator namin si Professor Jesusa Ortuste. But I think they focus on Arabica kasi di ba Arabica grows in the higher elevation. So actually dun sa bundok nakatangim, it's not in the campus that we got the coffee leaves, kundi doon sa bundok. Same then with Benguet State University. Sa parang bundok kami nagkolekta. In Cavite, the germplasm collection is within the campus. So probably you can contact, at least yun yung mga na-contact ko. So if you're interested, maybe you can contact them. Yun, and then maybe establish your collaboration or do further work, no? Sir Bernard, no? Oo.
Okay po, so thank you po, ma'am. From Sir Arnold Malbataan. So speaking of Barako po kanina, what Liberica it should be daw po ba? So according to Aaron Davis of KEW Botanical Garden ay meron daw pong 9 varieties. And yet possible magkakaiba yung lasa and physical characteristics po. So ano daw po ba talaga yung Barako po?
Actually, we are dependent on our collaborators. So, yung sinasabi sa amin na Barako, yun yung, nire-refer din namin na Barako. So, actually, yun nga, yung collaborators namin from Cavite State University, doon kami kumukuha nung, yun nga, yung leaf samples, no? Nung ina-identify nila na Barako. Although, sinamahan din nila kami doon sa isang coffee farm sa Amadeo, Cavite. And then, yun yung tinuturo nila sa amin na Barako coffee also. Same din dun sa, meron din kaming nakuhang sample from a farm in Lipa through the Philippine Coffee Board. Well, ang identification nila din sa amin is Barako or Liberica. So, as you were saying, maybe there are, but we have not distinguished, at least at the molecular level, pare-pareho yung nakikita namin na banding patterns. I think as you were saying, yung the taste may differ. This may be environmental also because we found from our collaborators sa CAFS, depende sa soil type, sa pH ng soil, ganon. And maybe other conditions, nakaka-affect siguro yung environment doon sa magiging expression nung aroma or taste of coffee. Actually, yung former PhD advisee ko who's also doing work now on coffee, yun ang kanyang pinopursue na research. She is studying yung mga terpenes na tinatawag because these have been associated nga with the taste and aroma. And baka yon kasi meron siya nakikita na iba-ibang mga terpenes na present sa iba-ibang varieties. So maybe Daisy can discuss that aspect of yung taste and aroma. Oo.
Very interesting po yun, no, yung sa aroma po.
Oo. That was Daisy's dissertation, yung mga volatile compounds from coffee. Yun, and she focused on Liberica kasi yun nga sana yung gusto nating i-develop also as kasi especially siya yung madaling itanim, especially here in our part sa Luzon, sa South.
Okay po, so may dagdag lang po si Dr. Fernando. So we'll have Dr. Miriam Baltasar of Cavite State University po next week in our coffee webinars. Maybe we can consult her po regarding some points mentioned by Dr. Cao po.
Yes, yes. Oo, kasi yan, sila yung nagme-maintain ng germplasm collection. So, they might be having other characterization kasi of the plants in
Okay
the
po.
germplasm collection.
Okay po, thank you po. So, from Mary, sorry po, hindi ko mabasa. Oxero. Good morning po. Are there results from the study on pollen morphometrics that implies its association to its potential to hybridization between different coffee varieties po?
Oo. Actually kasi, as far as I know, limited yung hybridization studies nga sa coffee dito sa Pilipinas, no? But yung close relationship again, no, nung Arabica at saka yung Canephora, ang isa naming nakita lang din sa pollen morphometrics, which is consistent dun sa mga molecular studies. And I think the reason is because, yun nga, Canephora is one of the parents of the Arabica. Kasi for those na siguro hindi familiar, Arabica is actually a hybrid of Coffea canephora and Coffea eugenoides. So dalawang diploid na nag-hybridize to produce the tetraploid Arabica. So I'm sure if you, kasi nagagawa rin nga kami ng genome studies, makikita yung close relationship nung genome din nung Arabica at saka Canephora. I think that's yun yung nakikita namin na close relationship from the pollen and even the other markers nga that we have studied.
Okay po. So from Keen Jade Asparin, good day. What are the plans and measures to improve the market of other varieties to compete with specialty coffee varieties?
Actually, syempre, from my end, parang laboratory based kami. But we wanted to come up nga with technologies that we can hopefully help farmers kasi sila yung directly no nagpa-plant ng coffee, and then aside from farmers yung because I learned may mga parang cooperatives, associations, yun nga yung Philippine Coffee Board, ang isa naming na-encounter, no, at other I think organizations para makatulong. Personally, ang thinking ko, maybe from our studies, yun nga, we can help recommend yung mga varieties na makikita na, let's say nga, resistant or okay yung taste nga, yung ginagawa ni Daisy, and even yung yield. Kaya lang, right now, yung concern nga yung other varieties, hindi pa namin naaaral actually yung other varieties. Yun nga, that are out there. As somebody was saying kanina, maraming baka varieties pa yung Liberica and so on. Kasi nga, syempre, from the academic point of view, nag-start kami dun sa characterized na muna yung mga NSIC registered varieties. And then, ngayon kami pumupunta nga sa field para mag-characterize ng ibang mga varieties. And that's why we're doing field testing also to check kung ano naman yung mga nakatanim. So, maybe kung matest nga ito, resistant din, then iyon naman ang pwedeng ma-recommend sa farmers.
Okay po. So thank you po. So we have a question, Dr. Kao, from FB po. So it's from Professor Teresita Borromeo. So did you also assess the antioxidants in the roasted coffee?
Hindi po, kasi actually yung nag-introduce sa akin dito sa coffee research, si Dr. Ruel Mojica of the Cavite State University. He was at UP Diliman as a local postdoc funded by the Bureau of Agricultural Research of the Department of Agriculture. Siya yung gumawa ng mga antioxidant activities of roasted coffee. So hindi namin ginawa yun kasi may work sila, Dr. Mojica, on that. Tinetest niya iba-ibang temperatures for roasting. Tapos ano yung temperature, ganun, may iba-ibang conditions na ng roasted coffee at saka yung iba-ibang varieties nga na magbibigay nung at the same time parang still good taste and aroma pero mataas din yung antioxidant activity. Kasi nga ang idea dito gusto natin na yung coffee ay maging healthy coffee din sana. Of course, I think ang general trend however na nakita niya, syempre the higher the roasting temperature, nasisira yung antioxidant compounds. So, hindi niya na nadidetect yung antioxidant activity. So, meron siyang nakuhang, nire-recommend niya na parang optimum roasting condition that is still tasty, kung ano ba, and at the same time may antioxidant compounds. Kinompare niya yung green coffee, kung familiar kayo, yung green pa lang yung seed, no? Yung bean, yun ang pinakamataas ang antioxidant compound actually na nakita. Pero kaya lang parang hindi nag-click yung green coffee kasi hindi pa syempre siya masarap because I think the aromatic compounds are not yet kung ano ba develop. So it's I think a balance between ngayon nare-retain pa rin yung antioxidant compounds pero excuse me, roasted siya. Roasted. So, ang ginawa namin, antioxidant compounds in the leaves. So, hindi sa roasting. Kasi, ang gusto ko naman na ma-come up na product sana ng farmers, yun nga parang coffee tea. Pwedeng gawin yung coffee leaves na tsaa. Yun naman yung, sanang isang product na pwedeng ma-market din ng coffee farmers. Yun.
Okay, ma'am, since nandun na tayo sa sa coffee tea. So meron pa tayong question from Marwin Magindayao. So do we have a project here in the Philippines aiming to commercialize production of coffee leaf drink? Yung coffee tea po natin.
I'm not aware of. Sana may mag-pick up yan from the younger researchers that we have here. Yan. Kasi nga technically retired na ako. So ito yung mga nagawa ko. I'm sure marami. Yun, that's why I included so many authors, yung mga dati kong RAs, dati kong thesis students. May mga nag-PhD pa doon sa authors din doon, si Nick. Maybe, and then our collaborators nga from the other institutions. Baka merong mag-propose na ng project. Yun kasi sana yung magiging offshoot dapat ng DA Bar project na may come up naman with a project on yun nga sana coffee tea production kasi yun mataas yung antioxidant activity niya, may antimicrobial activity rin siya as I have shown. Hindi ko na napakita yung results nga pala nung cytotoxicity test in the interest of time, but just to mention, we used MTT assay at wala kaming nakitang cytotoxicity. So we just tested for cytotoxicity, syempre just to make sure na hindi nakakalason naman yung gagawing tsaa. So maybe some here can pick up and come up with a project on that.
Baka po merong gusto dyan.
Oo.
Coffee. Para po sa mga ayaw ng brewed coffee, we have coffee tea. Ganun. Okay po. So from Menisa Antonio, meron po ba nagawang coffee flavor profiling on different species and varieties, ma'am? Meron po ba variability sa flavor profile nila?
Yan. That's the work of Dr. Daisy Santos. She did profiling of compounds found in coffee. So I will leave that to Daisy. Pero to answer the question, ginagawa, yes, meron. May publications, hindi ko na lang diniscuss kasi that is for Daisy to discuss na ng hindi mahaba din yung talk ko. That's part of her dissertation. Oo.
Okay po. So ma'am, meron po sa comment section, no? So curious daw po sa ano pong lasa ng coffee tea.
Actually, we have not tried yun nga. Kasi yun nga sana yung next phase, no, na kasi parang ang tiningnan namin, so may potential siyang antioxidant, antimicrobial, and then hindi siya cytotoxic, so pwede siyang inumin. Maybe the other yun nga researchers can, kasi I think dapat may collaborators yan sa food science, yan tamang-tama ang college to maybe do that, kasi syempre hindi namin expertise yung taste, di ba, yung mga ganan, quality, maybe another.
Opo, meron po tayong attendees from the food from IFST.
Oo, kasi yon. Kasi syempre gagawa, I'm sure may formulation dapat na gagawin, like anong concentration ng leaf sa tubig, yung mga ganon. So that could be ano po. Ayan, sorry, babasa ko lang po dun sa chat. Meron daw kaskara po ang ginagamit na panggawa ng coffee tea. So baka meron na ngang nagpa-practice ng using, I think from literature, parang I'm sure just like tea leaves or any other source of tea, pwede siya basta hindi naman nga siya toxic. So yan, thank you for the information po, Sir Matthew. Yan.
Okay po, so sabi po ni follow up po ni Sir Bernard Panabang, so meron daw po siyang fresh coffee leaves sa kanyang yard. Safe po ba i-try niya po ngayon?
Although ang ginawa namin doon sa study, doon sa cytotoxicity test, din-dry namin yung leaves. So we dried the leaves. Air-dried lang, nilagay namin sa dyaryo, nakalagay yung mga dahon. For about a month para dry na dry. Tapos pinulverize kasi namin. Yung ididikdik mo lang, either with a waring blender or yung manual grinding sa mortar and pestle. Tapos saka namin nilagyan ng tubig, yung ganon, ng solvent. Meron pa hong thank you sir Matthew, may additional information pa po siya nasa chat po. Yun daw peel and husk of the coffee cherry, no, ginagamit din. And taste notes po niya ay dependent sa coffee beans din na kinuhanan. Yeah, I'm sure kasi nga the beans are just within that husk. So, I'm sure yung mga substances ay nagdi-diffuse din doon. So, magre-reflect nga yung kung ano ang lasa.
Yes po. Okay po. So, from Annalie Solanya. So, good morning po, ma'am. May 4 clones of Robusta po kami dito sa station sa Surigao del Sur, but hindi po ako confident sa identity ng clone. Saan po pwedeng makapag-DNA identification and magkano po ang estimated payment per clone? Salamat po.
Well, I don't know kung merong gumagawa nung parang service to identify. We can pero actually kasi hindi kami kung ano ba sa service, we're not a service laboratory. So actually wala akong costing or whatever for research purposes yung ginagawa namin. Maybe you can check with CAFS, with Dr. Baltasar as the next. Baka meron silang services that they do, no? Or baka sa UPLB yan, baka may service lab or eventually may service laboratory. But unfortunately hindi kami nagsa-service kaya wala akong costing. We can always identify, pero wala kaming kasi nga service na ganon. So baka mas madali pagka yung may service laboratory. We can check with Dr. Baltasar next time, sa next speaker.
Okay po, ma'am. Thank you po. So, from Reynaldo Abrena, paano po masasabing pure coffee sa 100% refined?
Actually, yun nga yung I think may project ang CAFS dyan. Yun nga, pagka kunyari mga we don't know. Yun nga yung sinasabi ko kanina, yung assurance of consistency. Let's say, you want to know kung pure siya na, let's say, Arabica o kaya pure siya na blend, ganon. I think may gumagawa na yan na they can extract nga the DNA kasi DNA is very stable doon sa coffee, let's say granules or beans or whatever, kung anong sample meron. And then pwede mo ma-barcode and ma-check kung let's say ang gusto mo kasi ang after ka Arabica, or makikita mo nga ano pala siya, blend siya ng Arabica at saka let's say Liberica, ganon. I think CAFSU has come up with products na blends, yung cafe, ano yun, Aguinaldo coffee, naka-taste na ako. Pero ano daw nila, trade secret nila kung ano yung actual composition and percentage of, I think it's, well, sinasabi Arabica and Liberica, pero hindi, syempre trade secret yung percentage nung alin ang Arabica, ano, kasi that gives it the, syempre, the different flavor kung ano ba. But there is a way to check na yun nga, kung blend siya or pure siya na coffee.
Okay, papunta po ulit tayo sa Facebook questions. So, for review of from Allen Gala, for review of related lit po ng study nyo po for the tea coffee, saan po naka-publish for reference po? Saan daw po naka-publish yung sa coffee tea?
Hindi pa namin pinablish nga yung sa coffee tea because as I've said, we did not pursue going into the production of the coffee tea. We wanted, actually, yun nga, work namin yun ni Dr. Ruel Mojica, DA bar funded project. After his local postdoc nga sa UP, Diliman, sa NSRI, Natural Sciences Research Institute, he went back to CAFSU and did research projects on coffee. And then yun nga, he did the roasted coffee. I did the leaf samples for antioxidant activities. Pero I was thinking, yun nga, yung collaborators from CAFSU probably and the other institutions, kasi inintroduce din nila kami sa DA sa Visayas, nakapunta rin ako. Yun, I was thinking yung mga collaborators na ang gagawa ng production, kung ano ba the production side. So wala akong literature on yung kung may gumawa na. Parang wala yatang nag-pursue. Baka may gustong mag-pursue.
Sige po, ma'am. Tingnan po natin, baka po merong may gusto nga po dyan.
Oo, kasi hindi yung lab ko, hindi kami sa production side sa parang more of research side kami. We're banking on our collaborators na sila yung ano ng production side.
Okay po.
Yung mga commercialization. If ever, what I wanted eventually later to commercialize is that detection kit. Oo.
Okay po. So, ma'am, meron pong nagtatanong po ulit sa Facebook from Clyde Saniel. May I ask po on how to extract the toxicity po ng plant species? What standard method would you suggest? Because I am planning on conducting a research of native bean plants, but I found out that toxicity of the said plant is too high. Method, ma'am.
Actually, ano po yan depende po. Well, I'm not really a chemist, no? Pero from yung may collaborators din po kasi kami sa chemistry, yun nga yung sa mga aroma compounds, no, ng coffee. Pero kasi kung toxic, ang dami-dami kasing toxic may method yan. Maybe we can ask yung chemist talaga. I will not pretend na ano ako para dun sa extraction ng mga toxic compounds. Kasi honestly, hindi namin ginagawa yung extraction of toxic compounds. What I did, meron po kasing yung assay na sinasabi ko, so may extract kami, di ba, nung coffee leaves. Tapos, binibigay, ini-introduce namin yon sa cell line na tinatawag. May mga cell lines that are available. Tapos, yung MTT assay, parang may change din siya in color para malaman mo kung namatay yung cells o hindi. Parang ganun yung idea nung cytotoxicity test. Tapos, we can test that also in rats or mice. And then also in, let's say, mga brine shrimp assay, meron po din noon. Yun ang ginagawa namin, yung leaf extract, binibigay namin dun sa test animals or test cells. And then, ang findings lang namin, hindi sila namamatay. So, hindi na ako na interested na mag-extract ng toxic compounds kasi wala namang toxicity yung coffee. So, but other plants, I'm sure you know, may mga toxic compounds, di ba? Kaya may mga variety, let's say, ng cassava na nakakamatay, meron silang pinuproduce na toxic compound, ganon. At saka yung ibang halaman, di ba? Kaya hindi natin pwedeng kainin. Okay, namamatay yung mga insekto kasi may mga toxic compounds yung mga ibang halaman. So, I'm sure there's a method for that if you're interested in that. Nagkataon hindi kami kasi wala akong experience in extracting toxic compounds.
Okay po, so meron pa po bang questions for Dr. Cao from our audience? Wala na ba sa Facebook? Yes, last question po, ma'am. From Patricia Ann Alano. Good morning po, Dr. Cao. Question po tungkol sa kit. Since the kit is ideally for preventive purposes, is there a criteria for the plant that should be tested by the kit or other than physiological observations, like leaf rusting?
Actually, as I was mentioning earlier, the kit is yun nga preventive as you have said, para kasi kung meron ng manifestation ng rusting, kita nyo yung may mga dilaw-dilaw gold, yun nga parang gold yung itsura na bilog-bilog sa dahon, then of course you can say na ay may coffee leaf rust infection na itong halaman na ito. So parang at that point, hindi yun yung use ng kit namin. Parang ang use ng kit namin, yun nga, as you were saying, a preventive. Ang idea ko, let's say, may seedlings na idinidistribute ang Bureau of Plant Industry to coffee farmers, or baka may mga seedlings na available sa mga coffee farms. Parang itetest muna siya kaagad kung resistant siya o susceptible. And therefore, kung mag-turn out na present nga yung susceptibility gene, yung susceptibility band, ang ano namin is to advise, let's say, yung DA, kung sino nagdi-distribute, na wag niyong itanim yon. So, ang itatanim nyo, yung magta-turn out na walang susceptibility, yung may resistance genes. Then, yun yung itatanim. As I was saying, let's say, ang initial results namin among the NSIC registered varieties, yung red bourbon appears to be having resistance compared to yellow bourbon and caturra. So right now, what we're advising, let's say, or at least recommending, would be for DA or to kung ano mang mga associations to distribute yung variety na ito if you want to plant Arabica. Now, from our collaborators, let's say, sa CAFS, nung nag-sampling kami, yun nga, nakita namin according to Dr. Baltasar, while Liberica is supposed to be generally resistant, meron kami mga coffee leaf rust infected plants na na-observe, na kinuhanan namin ng leaves, sinample namin. It seems nagkakaroon na rin ng susceptibility yung Liberica. So maybe yun yung goal namin, to screen alin yung resistant. Baka may, yun nga, may mga resistant varieties din ang Liberica. Then, iyon yung ire-recommend na itatanim instead of yung makikita mo ng susceptible kahit wala pa nga yung infection. For example, remember in 2020, nagkaroon ng yung ashfall sa Taal before the pandemic came in March. By January, maraming farms, may lumapit sa amin na reporter, maraming coffee farms na nasira sa Batangas, na tabunan ng ash fall yung mga halaman nila. And therefore, ang sabi namin, if you will replant, then i-replant ninyo yung resistant varieties na. Kasi yung mga nakatanim, wala na tayo in a way magagawa. Except to practice yung mga pruning, di ba maraming mga practices din naman. Except, syempre, chemical pesticides, ayaw natin. So, ganun yung or if you're starting a farm, then you plant yung supposedly resistant. Yun yung pinaka-use nitong detection kit.
So, thank you po, Dr. Cao. So, I think that's the last question po. So, pasa ko na po ulit kay Ricky. Sir Ricky, thank you.
Yeah, thank you everyone
po.
for that very interactive, no, tsaka fruitful discussion that we had. So to show our gratitude and appreciation, join me in awarding this certificate of appreciation to Dr. Ernelea Picao. Allow me to read the citation. College of Agriculture and Food Science, University of the Philippines, Los Baños, Laguna, Philippines, present this certificate of appreciation to Ernelea Picao for serving as a resource person on the webinar entitled Development of Genetic Markers for Coffee Varieties Grown in the Philippines during the coffee webinar series held on 15th of November 2023 via Zoom. Given this 15th day of November 2023 at the College of Agriculture and Food Science at the University of the Philippines, Los Baños, Laguna, Philippines. Signed, Dr. Elpidio M. Maglisip Jr., Dean of the College of Agriculture and Food Science. Ayan. Maraming salamat po, ma'am, for sharing your expertise with us today. So don't forget to like and follow our official Facebook page. That's UPLB College of Agriculture and Food Science. And subscribe to our YouTube channel. That's CAFS UP Los Baños. And that brings us to the end of this episode. To formally end today's episode of the Coffee Webinar Series, may I call on Ms. Maria Lea H. Villavicencio, the Research and Extension Coordinator of the Institute of Plant Breeding, for her closing remarks.
Magandang umaga po uli sa ating lahat. To our speaker for today's seminar, Dr. Ernelea Cao, the coffee enthusiasts and coffee lovers that attended this webinar. Malapit na pong, ito na po ang end ng ating webinar series, and as we end this captivating topic on the world of coffee, we find ourselves tapos na. So hindi naman, but we hope that na marami kayong natutunan sa binigay na topic ni Ma'am Cao. Today, we delve into a topic of utmost importance, shedding light on the intricate world of coffee varieties and their genetic markers, specifically those thriving in the Philippines. In today's episode, we took a closer look at the heart of coffee diversity in the Philippines, exploring the development of genetic markers for different coffee varieties. This groundbreaking work not only deepens the understanding of genetic makeup of these beans, but also holds the promise of more resilient and sustainable future for coffee cultivation. The fusion of cutting-edge technology and traditional knowledge showcased today's underscores the importance of collaboration in pushing the boundaries coffee research. So nakita nyo naman na maraming involved sa research na nagawa ngayon ni Ma'am Ernelea Cao. As we reflect on the insights shared by our esteemed speaker, let us carry forward the spirit of curiosity and appreciation for the intricate nuances that make each cup of Filipino coffee a unique experience. The development of genetic markers not only empowers farmers and researchers, but also ensures the preservation of the rich coffee heritage that has been cultivated for generations. I extend my heartfelt gratitude to our speaker, ma'am, for generously sharing the expertise, and to each of you for your enthusiastic participation. Our collective journey through the world of coffee has been nothing short of inspiring, and I encourage you to explore and learn and, of course, savor the diverse flavors that this incredible beverage has to offer. As we part ways today, let us carry with us the knowledge gained, the connections forged, and renewed appreciation for the global symphony that is the coffee industry. May your future coffee experiences be as rich and diverse as the varieties we explored in this series. Maraming salamat at naway ang inyong bawat tasa ay puno ng pag-asa. Happy brewing, everyone!
Thank you very much, Ma'am Leia. So we'd like to hear your thoughts about today's episode. You may give your feedback by filling out this evaluation form. Just scan the QR code or go to the link flash in your screens. So here is our QR code. Just scan this or go to the link down below, the QR code. All right, and the coffee webinar series is just about to get more beautiful. Don't miss out the next episode where we will talk about identification and genetic characterization of Philippine coffee. We will be joined by Dr. Miriam D. Baltazar, the research director of the Cavite State University. This will be on the 21st of November next week, Tuesday, same time from 9 a.m. to 11 a.m. So on behalf of the College of Agriculture and Food Science, thank you for joining us today. I'm Ricky Brian Biagtan from the Institute of Plant Breeding, and this is the Coffee Webinar Series. See you on our next episode. Maraming salamat po.
Thank you
Thank you
po.
po, Ma'am Kao.
Thank you po, Dr. Kao.
Thank you very much also for inviting me, and thank you to everyone who attended for your participation. All the best po.
Sa inyo rin po, ma'am.
Thank you.
Ma'am,
Sige
bye-bye po.
Bye po.
Leave na po ako. Bye-bye. Take care, everyone. Nice to see you.